Hi everyone, and welcome to today's session. We've been having a series of protein metabolism, and so today we are going to look at catabolism, or the breakdown of proteins. So by the end of this session, we'll be able to describe protein degradation, catabolism of amino acid, and the fate of carbon skeleton and ammonia as generated by the breakdown of amino acids. So we start off with protein turnover, and as we all know, proteins within an organism are being continually synthesized and broken down. And this continuous synthesis and degradation of protein is known as protein turnover. Important to note, the rates of protein synthesis and degradation vary according to the physiological needs of the body. The rate of protein synthesis is high during growth, lactation, and also post-operative recovery, just to name a few, whereas in starvation, in cancer, and during morphogenesis, we see that the rate of protein degradation is very high. Now, the appearance of a given protein depends on its rate of degradation versus its rate of synthesis. If a protein has a fast rate synthesis and a slow degradation rate, it will increase in abundance within the cell. On the other hand, proteins that have both a swift or a fast degradation and synthesis have the fastest turnover rates. So let us look at protein half-life, and body proteins have lifetimes, which are expressed in terms of half-life. And this is the time it takes for the amount of a specific protein in a cell to be reduced by half, and this is done through degradation. And you find out that half-life of proteins range from minutes to years, and this is dictated by the needs of our cell. So it may either increase or decrease depending on cell needs. Now, some examples here of half-life of different proteins. We have the muscle proteins, which have a half-life of 160 days, whereas we have lactic dehydrogenase and cytochromes, which have less than 100 hours of half-life. So let's now look at protein breakdown or degradation. And it is not clear what triggers or initiates protein degradation. However, we have some structural features which serves as signal for the protein to be degraded. And these include ubiquitination, pest sequences, and N-terminal amino acids. So when you're talking about ubiquitination, we're talking about a small protein, that is ubiquitin, which is present in eukaryotes. And it serves as a signal for degradation of a particular protein. However, it is important to note that there has to be attachment of multiple ubiquitin molecules so that a protein is recognized and degraded by the proteasomes. A single ubiquitin molecule attached to a protein does not always signal degradation, but rather serves as a protein modification. When you're talking about pest, here we are talking a region rich in proline, glutamate, serine, and threonine will serve as a mark for degrading that particular protein. On the other hand, N-terminal amino acid, we're talking about presence of amino acid including arginine, lysine, phenylalanine, and aspartate, which are found at the end or at the amino terminal and serves as a signal for degrading that particular protein. So let us look at each of these signals or markers closely. We start with ubiquitination, and components of these pathways include ubiquitin, which you said it's a small protein consisting of about 76 amino acids. And it tags other proteins for degradation. You also have the proteasome, and this is just a large proteolytic complex that degrades the ubiquitinated proteins. That means it has enzymes that are involved in the breakdown or in the or in degrading the ubiquitinated proteins. Now let's look at the process of how ubiquitination takes place. So we start with activation. And here we have ubiquitin activating enzyme 1, and using an ATP dependent process, it forms a bond between the C-terminal carboxyl group of ubiquitin and the cysteine group of ubiquitin activating enzyme. And this is followed by conjugation, where we have E2 ubiquitin conjugating enzyme binding to the activated ubiquitin and E1 enzyme complex. And E2 will catalyze the transfer of ubiquitin from the E1 site to the active site on E2. And finally, we have ligation, where E3 ubiquitination ligase creates a peptide bond with the target protein's lysine and the ubiquitin C-terminal glycine. So now the protein is ubiquitinated. And so what is the fate of this protein? First, it might undergo degradation, where the protein is degraded by the proteasome. And the proteasome will unfold the protein and then breaks it down into small peptides and amino acid. The protein may also be subject to other processes, where we see that ubiquitination events do not always lead to degradation, but can also serve other signaling purposes, including DNA repair and also immune response. What's the clinical relevance of this pathway? So we see that the ubiquitin proteasome pathway is not just a cellular mechanism. It also has other significant role it plays clinically. First, in cancer, where we see that dysregulation of the ubiquitin proteasome pathway has been linked to various cancers. When this pathway is impaired, it will result in accumulation of defective proteins, which will contribute to tumor growth and also cancer progression. When it comes to the neurodegenerative diseases, we see that this pathway is very important in that an impairment of the ubiquitin proteasome machinery will lead to protein aggregation, which is a hallmark or a common hallmark of Parkinson and also other disorders. When it comes to infectious diseases, we see that there are some pathogens that can inhibit the ubiquitin proteasome pathway so that to avoid being degraded. And so this evasion mechanism allows them to escape being recognized and destroyed by the immune system. On to the pest sequences, and here we say that the pest sequences are defined by high content of proline, glutamic acid, serine, and threonine. And what is their role in degradation? They act as signals for rapid protein turnover. Therefore, removing or disrupting this sequence will result to a protein's half-life, or will increase a protein's half-life. While less adding the sequence will shorten half-life of a particular protein. So what is the pathway? How do they degrade, or how are they involved in the degradation of proteins? So they facilitate ubiquitination of a protein. This is where we have said is where a ubiquitin molecules tag or is attached to a protein and subjected to degradation. Some examples of proteins with pest sequence include the nuclear factors, the tumor suppressor, transcription factors, and also some research have also established that some proteins in the malaria parasites, that is the calmodulin, and other various immunogenic proteins have pest sequences. Now, when talking about the N-terminal pathway, we have what we call the N-end rule pathway, which basically describes how the identity of the N-terminal amino acid in the protein determines how long the protein will last inside the cell. What are we saying? Some N-terminal residues may either stabilize proteins, while others may target them for rapid degradation by the ubiquitin proteasome system. And how does this go about? As we all know, protein synthesis produces a polypeptide chain which contains a specific N-terminal residue, and often this is methionine. The polypeptide chain may further undergo post-translation modification, which include the cleavage of methionine or addition of acetyl groups, and therefore exposing a new terminal residue. And so the cells will be able to recognize the N-terminal residue either as a stabilizing or a destabilizing signal. If destabilizing residues are recognized by specific E3 ubiquitin ligases, they will tag the protein with ubiquitin for degradation by the proteasomes. And so here is a table just illustrating or showing some category of the N-terminal residues with the amino acids involved and what are the effects on protein stability. Now, for example, if we're talking about the stabilizing residues, here we have the likes of methionine, serine, the threonine, and if they are found at the N-terminal residues of a polypeptide chain or a protein, they confer long half-life to that particular protein and therefore giving stability to the protein. In case of destabilizing amino acid, these include arginine, phenylalanine, leucine, tyrosine. They confer short half-life and therefore decreasing protein stability. It's also important to look at some enzymes that are involved in protein degradation once the proteins are marked for degradation, and these include the protease and the peptidases. We have examples of proteases such as collagenase, elasticase, which now hydrolyzes proteins forming oligopeptides, that is many peptides. Now, the oligopeptides will undergo further degradation by peptidases to give rise to dipeptidase, which are then subjected further to degradation, and this is done by the action of dipeptidases to give rise to amino acids. So other enzymes involved include the megapain. We also have the proteasome, as we have looked at, and they possess multi-catalytic function, which are present in the cytosol of most of the mammalian cells. So what are the fate of these amino acids? Some of them may be used in the synthesis of structural proteins, functional proteins, some small peptides of biological importance, including glutathione and endorphins. And as we're going to look at, most of them can undergo catabolism to generate ammonia and alpha keto acids. So let us look at that in detail, the breakdown of amino acid. And this occurs in two main stages. We have one, removal of the amino group of amino acid as ammonia, which you will see that it's converted to urea and excreted as urine. And this involves 2 or 3 different ways of removing the alpha amino group. We have the transamination, which is closely followed by oxidative deamination. We also have oxidative deamination and thus finally non-oxidative deamination. The other main stage is the conversion of the carbon skeleton of the amino acid into intermediates of TCA cycle or the Krebs cycle and also acetyl-CoA. So we're going to look at these 2 stages in details. And so we start with the removal of the alpha amino group, which may be done through transamination. And this is the first step in the catabolism of most of the amino acid. Here, enzymes known as transaminases or aminotransferases are involved, and they're found both in the mitochondria and cytosol, and they require pyridoxal phosphate as a cofactor. So here we have an example of an amino group, and the alpha amino group is transferred to alpha ketoglutarate, which is then converted to glutamate. And the resulting product after the removal of the alpha amino group is an alpha keto acid, also known as the carbon skeleton. And as we have said, this is catalyzed by enzyme known as aminotransferases and require pyridoxal phosphate as a cofactor. So in most cases, we see that alpha ketoglutarate is the alpha amino group acceptor. And depending on the enzyme, most of these, depending on the amino acid, we name the enzyme as per the amino acid. So if this was aspartate, the name of this enzyme will have been aspartate aminotransferase. If it was alanine, it would have been alanine aminotransferases. So when you're talking about oxidative deamination, the formed glutamate after transamination is then transported to the liver where it undergoes oxidative deamination. And this process is characterized by glutamate dehydrogenase, where we have NAD+ peaking in presence of water, peaking 2 hydrogens. So they act as cofactors, so it can either be NAD+ or NADPH to give rise to alpha ketoglutarate and ammonia. We have the non-oxidative deamination, and here we see that some amino acids are characterized by specific enzymes. For instance, we have serine dehydratase, which is involved in a dehydration of serine to give us dehydroalanine, followed by the hydrolytic loss of ammonia. And as we see, it requires peroxophosphate as cofactor. So serine first converted to a hydroalanine and finally to pyruvate, generating ammonia. We have also cysteine desulfurhydrase, which is present in the bacteria and result to the conversion of cysteine to pyruvate, also releasing ammonia. So what are we seeing? The breakdown of this amino acid is generating a carbon skeleton and ammonia. There are also other amino acids which have amino group at their R or the side chain. And these include glutamine and asparagine. So how then do we remove the amino group at their side chains? And this is done by specific enzymes. For glutamine, we have glutaminase, which is able to remove the ammonia or the amino group as ammonia in presence of water to give us glutamate, whereas we have asparaginase, also present in the kidney, removing the amino group from the side chain of asparagine using water to give us aspartate. So let's first look at the fate of the carbon skeleton. This is what we have seen. Amino acids are broken down to release carbon skeleton and ammonia. And so let us look at the fate of this carbon skeleton that are generated from catabolism of amino acid. And we see that amino acid can be classified based on the metabolic fate of the carbon skeleton. It's either glucogenic, ketogenic, or both glucogenic and also ketogenic amino acid. So when you're talking about glucogenic amino acid, we're talking about carbon skeleton of this amino acid are converted to either glucose or intermediate of TCA cycle. And so these products may be pyruvate oxaloacetate, alpha ketoglutarate, succinate, and fumarate. And going by this diagram here, the glucogenic amino acids are presented by a pink shade. So we have, for example, phenylalanine and tyrosine. When they are broken down, their carbon skeleton will lead to formation of fumarate, which is an intermediate of TCA cycle. We also have glutamate, which is broken down, or when it is degraded, it leads to the formation of alpha ketoglutarate, an intermediate of the Krebs cycle. So examples include glycine alanine. All these are non-essential amino acids. So we can safely say that all non-essential amino acids are glucogenic. Now, when you're talking about the ketogenic amino acid, the carbon skeleton of these amino acids, they give rise to fat-like substance, or they are intermediates of fatty acid metabolism or catabolism. And the products may be either acetoacetyl-CoA and acetyl-CoA. So for instance, we have isoleucine, leucine, threonine. They give rise to acetyl-CoA. The same with lysine, tryptophan, they give rise to acetoacetyl-CoA. And so they are classified as ketogenic amino acids. Now, when you're talking about the both of them being glucogenic and ketogenic amino acid, we say that the carbon skeleton of these amino acids are converted to glucose or intermediates of the TCA cycle and also fat-like substances. And examples include isoleucine, phenylalanine, tyrosine, tryptophan, and leucine. So this is the fate of the carbon skeleton of amino acid after being degraded. So what about ammonia? Ammonia is toxic to the central nervous system. And symptoms associated with ammonia toxicity include blurred vision and tremors and may cause coma and death in severe cases. And so how is ammonia toxic, especially to the brain? First, the entry of ammonia into the brain leads to the formation of glutamate from alpha ketoglutarate. And therefore, this deprives or diminishes available alpha ketoglutarate in the brain. And this results to the impairment of the citric acid cycle, that's the Krebs cycle, diminishing ATP production. And so this will affect brain cell function. Ammonia, which enters the brain, is also used for glutamine synthesis. And this reaction, where we have conversion of glutamate to glutamine, requires ATP. And so if ammonia is converted to glutamine, it will result to depletion of cellular ATP, affecting cell function. Finally, glutamate is a neurotransmitter. And so its formation will result to overstimulation of nerve cells by glutamate formed from ammonia and also alpha ketoglutarate. And overstimulation will result to cell dysfunction. So what do we do with this ammonia? We see that ammonia is produced in tissues. It has to be transported to the liver, where it is converted to a lesser toxic water-soluble product, that is urea. So ammonia is transported in two forms, that is in glutamine and alanine. How is it transported as glutamine? So from the brain and other peripheral organs, we see except muscles, ammonia released is transported to the liver and kidney as amide nitrogen of glutamine. And here we have the enzyme glutamine synthetase, which catalyzes the conversion of glutamate, picks up the ammonia or the amino group, and it is converted to glutamine. And this reaction, as we had said earlier, requires ATP as the source of energy. So glutamate picks an amino or ammonia group, it's converted to glutamine, and then it's transported to the liver. Now, in muscles, we find that glutamine synthetase activity is low, and therefore ammonia released from the deamination of amino acid is transported in the form of alanine to the liver. And this is done through a cycle known as, or a pathway known as the glucose-alanine cycle. So we see that glutamate is formed in muscle cell by transamination. So we have glutamate formed from the breakdown of amino acid or the muscle protein, will result to the formation of glutamate. And then glutamate will then transfer its amino group to pyruvate, a product of glycolysis, to form alanine. And this reaction is catalyzed by alanine aminotransferases. It will give rise to alpha ketoglutarate. And then alanine will then pass into the blood and travels to the liver. And in the cytosol of the hepatocyte, alanine transfers its amino group to alpha ketoglutarate to form glutamate and pyruvate. And this reaction is catalyzed by alanine aminotransferases. And so pyruvate through gluconeogenesis is converted to glucose, which is then transported via blood to muscle. So what becomes of glutamate? So glutamate can then enter the mitochondria, where we have glutamate dehydrogenase reaction releasing ammonia, which then through the urea cycle is converted to urea. So what you said is that apart from the muscles, all other tissues are able to transport ammonia as glutamine, whereas from the muscles, ammonia is transported as alanine. So let us look at the urea cycle. And when you're talking about urea cycle, this occurs in the liver, and it occurs both in the mitochondria and also the cytosol. And we have the first step, which is the formation of carbonyl phosphate, and it's a condensation reaction of ammonia, bicarbonate, and 2 ATP molecules. So when these three compounds combine, they form carbonyl phosphate. And this reaction is characterized by carbonyl phosphate synthetase 1. And you have seen it requires energy in form of ATP. And this enzyme requires N-acetylglutamate, which is synthesized from acetyl-CoA and glutamate in the liver, and requires magnesium ions. So the first step, as we said, is a condensation of ammonia, a bicarbonate, and 2 ATPs to give us carbonyl phosphate. Carbonyl phosphate is then condensed with ornithine to give rise to citrulline. And this reaction is catalyzed by ornithine transcarbamylase. Citrulline is then transported from the mitochondria to the cytosol, where it combines or it condenses with aspartate in presence of ATP to give rise to adenylosuccinate. And this reaction is catalyzed by adenylosuccinate synthase. And formation of adenylosuccinate is then formation of adenylosuccinate, as we said, it's an ATP-dependent process. It is then cleaved to give rise to fumarate and arginine. And this reaction is characterized by adenylosuccinase. The next step is a regeneration of ornithine, which is also related to formation of urea from arginine. So we have regeneration of ornithine, and it is coupled with formation of urea from arginine. Here we see in presence of water, arginine will generate urea, and it's converted to ornithine. And this reaction is catalyzed by an enzyme known as arginase. So we will appreciate that urea cycle occurs in the first two steps occur in the mitochondria, and citrulline is then transported to the cytosol, where through various intermediaries, it's converted to urea and ornithine, which will come back to the mitochondria. So we can summarize that reaction as ammonia plus a bicarbonate plus aspartate and ATP molecules to generate urea fumarate, which may enter the Krebs cycle, ADPs, and also inorganic phosphate. Urea cycle is also subject to regulation, and here we have the enzyme carbonyl phosphate synthetase 1, which is the rate limiting step, or it's a which catalyzes the committed step in urea synthesis. And we see that N-acetylglutamate, it's an activator of this enzyme. Here we see high protein will lead to high levels of N-acetylglutamate formation. And in most cases, we find that high protein will influence urea formation. For instance, during starvation, the rate of urea synthesis is also high due to the increased degradation of proteins, which will result to high levels of amino acid. And when these amino acids are broken down, they will release carbon skeleton and ammonia. So we can see that high protein will influence the rate of urea formation. Let's look at the diseases that are associated with the urea cycle. We have hyperammonemia type 1, type 2, citrullinemia, aciduria, and hyperaginemia. So for hyperammonemia type 1, it's due to the deficiency of carboxyl phosphate synthetase 1, which will result to metal retardation. We have hyperammonemia type 2. It's a common disorder, and this is due to the deficiency of ornithine transcarbamylase. And as a result, we have accumulation of carboxyl phosphate, which may be diverted to pyrimidine formation. Citrullinemia is due to the absence of enzyme adenosine synthetase, and we see that citrulline accumulates in blood and is excreted in the urine. So in this other disorder, we see it's due to the absence of adenosinease, and we see that adenosineate accumulates in blood and is also excreted in urine. Finally, we have the hyperaginemia. It's due to low agenase activity, and adenine accumulates and it's also excreted in urine. However, we'll see that some urea may be excreted in urine due to the kidney agenase. And so with that, we've come to the end of this session where we have looked at the breakdown or degradation of proteins to amino acid, and we have seen the fate of the products of amino acid, that is the carbon skeleton and ammonia, where we have seen the carbon skeleton can either be glucogenic or ketogenic depending on their products. The glucogenic are the ones involved in the formation of glucose, whereas the ketogenic will form the fat-like substance. You have also seen that ammonia, which is generated, also is very toxic and therefore must be converted to a less toxic compound, which is soluble in water, and that is urea, which is now excreted in the kidney, in the urine. And so in the next session, we're going to look at how do we synthesize non-essential amino acid. Thank you.